because I was at Okay, thanks for coming out, everybody. After Memorial Day special seminar, appreciate people coming out and attending today. So it's a pleasure to introduce today's speaker, Dr. Richard Gomer, who's professor of biology at Texas A&M University. Dr. Gomer received his Ph.D. in biology from Caltech, then did postdoctoral training at the University of California, San Diego. And then he joined the faculty in Houston at Rice University, where he became a professor of biochemistry and cell biology, and also held, and I think still holds, an adjunct appointment at Baylor College of Medicine. Is that right? Or not so much. Okay. He did for quite a while in the Department of Cell Biology, Bert O'Malley's department there, and had an affiliation while I was across the street at Rice. During his time at Rice, he also was awarded, received Howard Hughes Medical Institute investigator status that he renewed at least a couple times, which is no easy feat, and did very well during that period, obviously. He joined the biology department at Texas A&M in 2010. He serves on a variety of editorial boards and still does advances in molecular imaging, International Journal of Cell Biology. He's a member of the board and the faculty of 1000 and serves on the ed board of Biomedical Research International. He's also currently serving on an NIH study section on the lung injury repair and remodeling study section. And as indicated here, he's co-founded a company, ProMeteor, a clinical stage biotech company in development of targeted therapeutics to treat fibrotic diseases. And they're, based on what I could see at the website, looks like they're doing some very interesting things, have a lead compound and trials running and so forth. Dr. Gomer has worked on tissue size regulation, cell composition, and fibrosing diseases, working towards translating discoveries from basic science into the clinic. He's done a series of studies on regulation of size of tissues and tumors as well, and how this can be manipulated for therapeutic purposes. He makes use of dictyostelium as a model organism system. And DICTY, as many of you know, has been used for all kinds of really groundbreaking work in understanding signaling, cellular communication, and so forth. And he has, as I said, made great use of that, making a lot of strides. He takes a computational modeling approach in parallel with his biochemical and cell biology approaches. Using that approach, they've discovered a secreted protein and also the role of polyphosphate signals operating in negative feedback loops that can inhibit cell proliferation in DICTY, and are also looking into the signaling mechanisms of that to understand similar mechanisms in humans as well. They've made discoveries on human blood protein, particularly serum amyloid P, how it can regulate key steps in formation of scar tissue, as well as the formation of scar-like lesions and fibrosing disorders like congestive heart failure and pulmonary fibrosis as well. So I think it's pretty obvious that his work is fundamental, strong, basic cell biology, but has very important implications for a variety of clinical disorders and has a number of touch points to several of the groups represented here. So I look forward to hearing about your work, Richard. Welcome.
Thank you. So just before I get going, I'm a co-founder of and I have equity in a company called ProMeteor. So anything I say about this serum amyloid P protein and its use as a therapeutic, please take that with a large grain of salt. So as a basic outline of the talk, I'm going to tell you about a cell type called a fibrocyte that differentiates from monocytes. I'll tell you that there's a protein called SAP that inhibits this differentiation. SAP's a possible therapeutic for fibrosis. By looking at the SAP signal transduction, we found a second generation set of therapeutics. And then finally, I'll tell you how by looking at what SAP is doing, we found yet sort of a third generation of potential therapeutics for fibrosing diseases. So as an introduction, the main evolutionary driver behind fibrosis is the need to be able to heal wounds quickly. So if you have a dermal wound, as illustrated here, and there's fibroblasts at the wound edge, the fibroblasts will start proliferating to try and fill the wound in with granulation tissue. But we've evolved this really delightful and useful mechanism where monocytes, for instance, will extravasate from the blood vessels into the wound bed. And and they'll differentiate into these long contractile cells called fibrocytes. And the fibrocytes will actually help pull the wound closed. The other nice thing about this is you need to fill in all of this with granulation tissue before you can lay down a layer of epithelium, and you can start bringing in grams of cells from the spleen, from the circulation, from the bone marrow, and start dumping it into the wound bed to accelerate granulation tissue formation. So the punchline so far is that there are these cells called fibrocytes. They differentiate from monocytes, and they play a very good role in wound healing, helping form granulation tissue. Now, fibrocytes have sort of a multiplicative effect on wound healing. So fibrocytes will secrete factors that you may have heard of called things like TNF-alpha and TGF-beta that will activate resident fibroblasts. And they'll tell the local fibroblasts, hey, you need to start dividing, and you need to start making extracellular matrix proteins such as collagen and fibronectin. So they're sort of like these special forces troops. You can parachute a couple of them into some area, and these guys will speak fluent Gnorgnok and get hundreds of Gnorgnoks to attack the bad guys. So they have this multiplicative effect. Now, unfortunately, there's a whole series of diseases, about 62 of them, where you get something that looks like a healing wound, but inappropriately in an internal organ. So, for instance, this is what a normal human lung looks like. This is a human lung where the patient has probably inhaled little bits of particulate matter, coal dust, silica dust, and the body has made this terrible and stupid mistake. It thinks this is a wound and starts trying to heal this perceived wound, and the whole lung will start filling up with scar tissue. And if you look at the cells, it's very, very similar to a healing dermal wound. And there are lots and lots of different fibrosing diseases. The biggest, baddest one is congestive heart failure or congestive cardiomyopathy. And you can see this in the liver and in the kidneys. There's radiation-induced fibrosis. If we could shut down fibrosis, the radiation oncologists have found that if a patient has a tumor, they can kill that tumor, but they need to use such a high dose of radiation that it will cause, can anybody guess? Fibrosis, okay. So if you could prevent the fibrosis, the radiation oncologists could have lots and lots of fun curing cancer. And these are, if you integrate these, it's about 30 to 45% of deaths in the U.S. Cancer is about 25%. So you've probably heard that heart disease alone kills more people than cancer. So these fibrocytes, you see them in the fibrotic lesions as well as healing wounds, and they play a good role, but they also play a bad role in the fibrosing diseases. But nothing, when we got into this by accident, nothing was known about what regulates this differentiation, because you never ever see these cells in the blood, and you never or very rarely see them in a normal tissue. But in the wound or fibrotic lesion, something is triggering this. So what I'm going to tell you next is that we found by accident that there's a normal human blood protein that inhibits this, and this can be then used as the basis for a first-generation therapeutic. So you've probably all seen pictures of white blood cells in culture, and they look like these little bitty round cells. But when people culture blood, they always have some serum there, usually 1%, 10%, but you can see this even with 0.1% serum. For reasons I won't go into, we decided to start looking at proteins secreted by white blood cells, and I wanted to have human white blood cells in serum-free medium so we could collect stuff they were secreting. And we were checking on the cells to make sure they were okay, and to our shock and amazement, we saw in 3 days these incredible long, skinny cells appearing. And I did video microscopy, and they're motile, so this isn't some weird artifact. And people had been able to identify fibrocytes and show that they stain for combinations of hematopoietic markers such as CD45 and collagen or fibronectin. So these cells stained double positive and so appeared to be fibronectin. But if you take this as the ground state and you take this as the perturbation, that means these cells can normally appear, and there's something in serum that inhibits this. That makes sense? So once I hit cruising altitude, I might start going fast, so please stop me at any time. Any questions so far? Okay. So we got a bottle of serum and started fractionating it, and the factor in the serum looked like it was big. It was a protein, and we're doing lots and lots of protein purification of rare secreted proteins in, for instance, dictyostelium. So we saw we were able to purify this to a single band using just a variety of protein purification procedures. And we did amino acid sequencing, and turns out this factor in serum that's inhibiting the differentiation of the fibrocyte is this protein called serum amyloid P. It's made by the liver, it's secreted into the blood, it's got about a 24-hour half-life in the plasma, but once it gets into the tissue, it's got a long half-life. And there's a variety of concentrations in people. So if you get individual people and say, well, how much sap do you have? The average is about 30 micrograms per mil, which is quite a lot. But there are some people with very low levels of sap, and some people in this room probably who have high levels of sap. And, you've probably heard of, this just appeared. You've probably heard of proteins called serum amyloid A or amyloid precursor protein, amyloid beta. Those are bad proteins, whereas this sap protein is a good protein. I need to make that careful distinction. And sap knockout mice are viable, but just the slightest little trigger will cause them to have rampant fibrosis. So here's the lungs from a wild-type mouse. Just you shoot a little saline or untreated, and you can see lots and lots of air spaces. Sap knockout mice, here the red staining is for collagen. And there's a little bit more collagen, but you give them a tiny little dose of an irritant called bleomycin that will trigger formation of reactive oxygen species and then fibrosis in a couple of weeks. Here, wild-type mice are getting a little bit of fibrosis. You can see this enhanced collagen staining. The sap knockout, way, way, way more collagen. So one of the normal functions appears to actually to limit fibrosis. And if we look at, for instance, in patients with pulmonary fibrosis, so around here it's probably called black lung, where the coal dust induces pulmonary fibrosis. The patients tend to have lower levels of sap. And if you parse out all these patients by sap concentration on one axis and the air capacity of the lungs on the other axis, so you have the patient take a deep breath and they blow into a tube and you measure how much air they can blow. That's the forced vital capacity. The patients with low levels of sap tend to have very poor lung function. Patients with higher levels of sap tend to have better lung function. And this has now been observed in patients with renal fibrosis, a weird fibrosing disease called scleroderma, and a bone marrow fibrosis called myelofibrosis. And we can use this sap differentiation, the effect of sap on fibrocyte differentiation to look at other things. So, for instance, it's been known that high-salt diet seems to potentiate heart disease, cardiac fibrosis. Now, what we see is that if you culture cells with a little bit of extra sodium chloride in there, the dose-response curve for sap is shifted. So to be able to completely inhibit fibrocyte differentiation, you need more sap. And at some given sap concentration that you might have, say, in a tissue, you see more fibrocyte differentiation when there's high salt present. And that's just a little aside. Any questions?
That experiment?
Pardon?
What was the duration
Oh, this was in culture, so this was 3 days. So we've actually published a whole paper on this, so we can parse this out as to, for how long do the cells need to be exposed to salt. And it's a fairly short exposure time. So you expose them to salt, and then wash it out, and let them go, and they still have this enhanced differentiation. If you're interested in, we've also done this in mice on high and low. So this predicted that high salt diet, low salt diet would affect fibrosis, and we published a paper on it, and I can walk you through that if you're at all interested in this. So let me go through this idea that SAP then, by inhibiting fibrocyte differentiation, might be a potential therapeutic for fibrosis. So I've showed you that SAP inhibits this differentiation. So if you could remove SAP, you might be able to drive this reaction to the right, and we actually found materials, like bandage materials effectively. You put on a wound, and it will pull the sap out of the wound. So any of you fallen off a bicycle or a skateboard, you all look like skateboarders to me, and gotten this road rash and all this yellowish gooey stuff on there. You've seen that? So that's serum from the blood clotting. And if you can, it's full of this fibrocyte differentiation inhibitor. So you could pull that out and leave all the nutrient goodies in there. You ought to be able to speed wound healing, right? So we made a bandage. It pulls the sap out, leaves everything else, and speeds wound healing. And we were completely unable to get any of the bandage companies interested in this. And finally, someone said, "Look, the bandage companies, they're in business to sell bandages. So if they're sitting there smoking their cigars and drinking their cognac, and some snot, knocks on the door and says, 'Hey, I figured out a way in which you could sell fewer bandages,' Just like, no, get out of here." So, but on the other hand, there's no treatments for fibrosis. So if you get a patient extra sap, we might be able to inhibit fibrosis. So we tried this in a rat model. So this is micrographs of the lungs, and this is a very, very standard model. There's a drug called bleomycin. It's a frontline chemotherapeutic for testicular cancer, but if it's given to patients in too high a dose, it'll cause pulmonary fibrosis. So you can actually shoot this into the lungs of rats or mice, and in 14 days, 21 days, you see all this scar tissue in the lungs. Here's what a normal-looking lung looks like. And if we do the bleo and then treat with SAP, the lungs look normal. Here's staining, this is H&E staining for collagen, do immunofluorescence staining for collagen. And there's a marker for some of the really aggressive fibroblasts called myofibroblasts called alpha smooth muscle actin. You see a lot here, but it's down to normal levels with the SAP treatment. And we can also use this not in just a prophylactic setting, but in a therapeutic setting. So for instance, here, we're looking at the blood oxygen concentration. So for control mice or people, it's around 97, 98. With bleo, the lungs are full of scar tissue. They can't get as much oxygen into the blood. The pulse ox is down. And if we do the same bleo, and the pictures I was showing you previously was all this collagen in the lungs at day 14. So we start injecting with SAP at, say, day 14, at 15, 16, all the way through day 20. Sacrifice at day 21. Pulse ox is back. The histology is back to normal. So this will reverse an existing fibrosis if the fibrosis is at a reasonably early stage. What we've seen is that these fibrocytes appear, and I can do video microscopy for many days. You just put a microscope in an incubator, and then the fibrocytes round up. And so if any of you had a scar, like I've got a scar here, it was heavy and thick, and then, after a few months, it got to be more softer. So the fibrocytes will sort of go away, the scar will go away. So if you prevent the formation of new scar coming in, you can just let the existing process of scar tissue being effectively resorbed sort of resolve things. And one of the questions was, why don't you see fibrocytes appearing in a tissue? So we found that if you've quiescent fibroblasts, like ordinary fibroblasts from ordinary tissue, they secrete a molecule, a protein called SLT2, and that inhibits this differentiation. And SAP, I just showed you, is inhibiting this differentiation. But SAP is also, if any of you are into innate immunity, SAP inhibits the differentiation of both M1 and M2A macrophages. So these are classically activated macrophages. These are profibrotic macrophages. And SAP seems to push monocytes to becoming mReg macrophages. And mReg macrophages sort of come into a situation and say, okay, let's everybody calm down. And they sort of can calm down either M1 or M2 type inflammation. And SAP will also cause M1 or M2A macrophages to become one of these mReg or M2C macrophages for the cognoscenti. Now I promised I would, or the title of the talk was a little bit about positive feedback loops. So I don't have time to go through the whole details of this. I showed you data from a colleague where fibrocytes secrete TNF-alpha and TGF-beta to activate fibroblasts, and we found that fibroblasts that have been activated in this way will secrete a basement membrane component called lumacan, and the lumacan will potentiate fibrocyte differentiation. So here we have one positive feedback loop that seems to be driving this fibrosis. And SAP, thankfully, seems to be dominant over this process. So even when there's fibroblasts present, making lumacan and fibrocytes, goosing the fibroblasts with TGF-beta and TNF-alpha, SAP can come in and arrest this process. Any questions at this point? We good? So we started a little company called Biomedical, and if anybody wants over a beer, I can tell you how we came up with that name. And we tried SAP, or we got various people, experts in this or that fibrosing disease, to try it out in their model system, and it works in ocular fibrosis, other models of pulmonary fibrosis, renal fibrosis, liver fibrosis. We collaborated with a guy to show that SAP inhibits cardiac fibrosis, and we found that SAP slightly reduces scar formation, slows it down. And then the company got two different experts in radiation-induced fibrosis to show that SAP will inhibit radiation-induced fibrosis. So the company then raised about $76 million in venture capital funding. They were able to make recombinant SAP in CHO cells at about a gram per liter. And they did a phase 1 trial, and it did just fine because it's a normal human protein. And the results of the two phase 2 trials completed enrollment in December of 16 after the $76 million in VC funding. Bristol Myers Squibb came in and paid $150 million as option payment to be able to buy the company for about $1.25 billion. So that $150 million funded 2 clinical trials, one for pulmonary fibrosis and the other for this bone marrow fibrosis called myelofibrosis that I mentioned. And 9 days ago, the results of the clinical trial were announced at the American Thoracic Society. So this is time on this axis. This is the for patient of any given age or weight. This is the change in their lung function, the FEV, the ability to blow out a volume of air. And untreated patients tend to decline pretty quickly. This blue is the 90% confidence interval for patients on current standard of care. There are drugs called pirfenidone and nintedanib, and patients are put on that, and that does slow the decline in lung function. And patients on the standard of care plus that SAP protein that I was telling you about, their decline is much slower. And this decline is starting to approach the decline you just see in normal people. But if we parse this out, this decline is much less. You see much less of a decline if the patients aren't on standard of care. The other.
Can I just ask a
Yes.
question about
Yes.
So going back to the previous slide,
Yeah.
two questions. Who were these patients in terms of the severity of pre-existing
They were.
FVC or?
They were. I can pull you out the data, but they were in pretty bad shape. These were patients.
They were less than 1 liter in terms of forced vital capacity, which would be quite severe. Or if they
I don't think they were that bad. They were between, I think, 75 and 50% of predicted.
Okay.
So that would be probably more than a liter.
And these percentages, 1 to 5%, do you know what that is in absolute numbers? Liters?
No, so it depends on the person. So you say, okay, here I've got a person, you know, of this size and body mass, and so this is the predicted, you figure out what the predicted FVC should be, and then you say, okay, what's their forced vital capacity at T equals 0, which would be up here, and then what's the decline in that percent predicted.
Well, it says from baseline, so I assume
That's-
that
yeah, well,
means
from-
each
yeah.
individual person's baseline.
Yeah, yeah. Yes, you're right. Okay, so this is from the time that they were started on that. Other questions?
How did you select the once a month for the trial and the frequency of administration?
So there was a phase 1B. There was a phase 1 that was single ascending dose for toxicity, and then they did a phase 1B on about a dozen patients. So about 4 were put on once a week, and 4 were put on once a month, and then different dose regimens. And 5 and 10 mg per kg once a month seemed to do as well as anything else. So they said, oh, let's do 10 mg per kg once a month. Then there's another test for lung function, which is even simpler to understand. So 6-minute walk test. So the patient just, you measure how far they can walk for 6 minutes. And the complete, I'm sorry, I was mixing up percent predicted and that change from baseline. This is the first time I've actually shown this data. It just came out. So actually what happened was at the Thoracic Society meeting, the MD presenting the clinical trial, at the very end of his talk said, this has now just come out in JAMA. And then there was an editorial accompanying it. So if you're interested in the gritty details, that's in the JAMA paper. So this is the 6-minute walk test. And so if you look at let's see, this is the change in from week 0 to week 28, and this is just directly in meters. So all the patients, there's a decline. The patients, whether or not they're on drug, the other drugs, there really wasn't hardly any decline. Patients not receiving the standard of care, which is either pirfenidone and nintedanib, there's a decline. This has been seen in much larger trials. But with that SAP protein, they're actually doing maybe a little bit better. And when they're actually on the drug, there's a little bit of a decrease. So, I mean, you have to take this with a grain of salt, but there's a possibility that they would do better on SAP alone versus SAP plus standard of care. But in any case, this is a reasonably large trial, so 39 patients in the placebo group, which includes either not receiving or receiving standard of care, and 77 in the getting the SAP. And so there's a roughly equal sized trial for myelofibrosis It completed enrollment at about the same time, but that was a 9-month trial. This was a 6-month trial. Analyzing the data for myelofibrosis is trickier because it involves getting bone marrow biopsies, but hopefully that data will be coming out pretty soon. I have my fingers crossed on that. There was about a 20-patient phase 2A trial, and those patients actually did pretty well. So this is bone marrow biopsies. So from a couple of patients. So this is at screening, and then this is after some amount of SAP treatment, and you can see all this fibrosis, and it's getting reduced. And if you look at computerized image analysis, most of the patients seem to be getting a little bit better, and just 1 or 2 patients seem to be getting worse. So I have my hopes up for these patients. Are there any questions at this point? I'm sorry. Yeah.
That's okay. Was the data for the forced vital capacity split up in the same way that this was, looking at patients who were
Yes.
or were not, and were the trends the same? Or
It wasn't as dramatic, but that's all in the JAMA paper. One of the funny things was, let me see, how did it work? If you looked at the controls looked funny for the forced vital capacity. So it was sort of like on drug, there was a reduction, and not on drug, it looked a lot better. So something seemed a little bit odd. But these have been patients who had were quite sick. A lot of the trials are for a much shorter amount of time, and they do not take patients as sick as these patients. So that might have affected the results of this trial. I'm not a pulmonologist, so I'm more of a dicty person. Yeah?
Is there any plans to measure diffusion capacity of the lung rather than forced vital capacity?
So they did DLCO, and that data was not analyzed centrally, but there wasn't much of a change. This was puzzling. There was not much of a change in DLCO.
The first 2 drugs were criticized for using FVC as a measure. When they were first released, there was some discussion of whether that was appropriate.
Right. So if you read the editorial in JAMA, which is written by 2 MD pulmonologists, one of the things they point out is that maybe it's the 6-minute walk test that's the best thing that, say, correlates with morbidity, mortality, quality of life, rather than the FVC. I would have thought the FVC, or both FVC and FVC1, would be pretty good measures, but the pulmonologists, some of them think that this 6-minute walk test is better.
That's usually the endpoint of that. It's usually shortness of breath.
Yeah.
And there's a lot of factors that go into that
Yes.
beyond the state of the lung. Although the correlation of shortness of breath and fibrosis is very good, but just wondered why transfer factor is not being used.
Transfer.
Sorry, the diffusion capacity.
Oh, the diffusion capacity. They did measure the diffusion capacity, and they didn't see much of a change.
Right.
So here are these patients that are declining significantly. This 31 meters in their 6-minute walk. Basically, these guys declined by half a meter, so instinctually from no decline. But the DLCO didn't change. So it might be that these patients were so sick that they had so much fibrosis already that you're not going to see much of an effect on DLCO. I don't get it. I mean, I'm just waving my hands and babbling, obviously.
On some of these trials, we don't look for when it has an improvement in DLCO, we look for a lack of worsening.
Mm-hmm.
So if you had over a 28 period no worsening, that could be interpreted as a positive.
Oh, there was no difference between any of these, this, or this, in DLCO.
Because many times you just look
So this has slowed the progression, and certainly for this 6-minute walk test, it's done some good things for these people. But we were taking, I'll reiterate, the patients who are in really bad shape. So several years post-diagnosis. A lot of the other trials, they'll take people who have just a tiny, tiny little bit of incipient fibrosis, you know, where the percent predicted is down by 10, 20%, you know, nothing too serious. But where somebody's done high-resolution CT, and you can see some honeycombing here and there.
What was the average age or time after diagnosis for the patients?
Oh, this is
You said
about,
several years.
I think 3.5 years. It was pretty bad. Other questions? But I would urge, since I've just seen this, because you know I was kept in the dark along with everybody else, but everything is in there. Any other questions? Okay, so let me tell you now a little bit about a second-generation version of SAP. So we were looking at the protein and asking, well, I wonder what domains on the SAP are actually activating the receptor. And SAP is actually structurally and has sequence similarity to a protein you've probably heard of called C-reactive carb protein, which has been associated with things like heart attacks. But SAP is I would say a good protein, inhibits fibrosis and fibrocyte differentiation, is anti-inflammatory. CRP seems to promote fibrosis, has no effect on fibrocyte differentiation, and it seems to be pro-inflammatory. And so we mutated amino acids on the surface of SAP that were different from the corresponding amino acids on CRP. And we didn't see really any difference in the ability of the mutant SAPs to inhibit fibrocyte differentiation. And then we're scratching our head and we realized, well, SAP's glycosylated and CRP isn't. Maybe it's the glycosylation. So the terminal sugar on SAP is sialic acid. And if we desilate SAP enzymatically, all of a sudden, completely loses its activity. Here's the curve for SAP. Yes, sir.
I'm just curious if it's
Oh, yes, ma'am.
functioning actually as
I don't have my glasses
in
on.
the body. And
What's
what's the biological significance of it?
the biological significance of what?
Of it being is it functioning as a pentamer in the body? And what's the biological
Oh, it's always a pentamer. So one of the receptors it binds to is Fc gamma receptor. An Fc gamma receptor binds the Fc portion of immunoglobulins. And you've got lots and lots of immunoglobulin in your body. And as long as the immunoglobulin isn't, I need a prop here, isn't cross-linked to something, it doesn't do anything to the receptor. But if I have a bacterium that's covered with immunoglobulin, and the immunoglobulin is sort of stable, and the Fc portions are hanging down from the bacteria, that grouping of non-mobile Fc domains will effectively signal to the Fc gamma receptor saying, oh, you're not binding any immunoglobulin, you're binding an immunoglobulin that is stuck onto something, because you'll see one of your buddy receptors right next to you, and they can't move. So you're looking at a bacterium that's, say, covered with bacteria. With me so far?
Yeah, but I don't know why
Okay.
it's a pentamers.
Okay, we're going to get to that.
That's my question.
The only time you don't want to ever heal a wound is if the wound is septic, okay? You do not want these cells closing a septic wound. Any of you ever heard of something called gangrene? Okay, so you can get gangrene if you close a septic wound. So as long as there's bacteria and stuff covered with immunoglobulin, that tells these monocytes, do not become a fibrocyte. You just go become a macrophage and start eating bacteria. Only when the wound is clean, then you can do it. SAP is mimicking that cross-link immunoglobulin to some extent for the FC receptor. That make sense?
Yeah, but I don't see why it has to be fivefold to do that.
Oh, yeah, why fivefold instead of 6 or 4?
Yeah.
Oh, yeah, no. It's just, yeah, no idea. It's just some fluke of nature or something. It's an incredibly stable protein. You can boil it, and it still maintains activity. But some of the key signaling appears to occur through the sialic acid. We desilate it, and it loses activity. We can take this other protein called CRP, which has no effect on fibrocyte differentiation. We mutate it so it's got an asparagine that'll get glycosylated. We can see that it's glycosylated, and now it will start inhibiting fibrocyte differentiation. So we searched around for a possible receptor, and it turns out there's a receptor that's expressed on innate immune system cells called DC sign that looks like it can bind carbohydrates. And so we can express this on HEC293 cells. And we see that SAP will now bind to those cells. CRP won't bind, desilated SAP won't bind, and of course, if you don't express it, and we did all the controls, you don't see any binding. So it looks like SAP will bind to this receptor called DC sign. We can use antibodies against DC sign to inhibit fibrocyte differentiation, and it's not working through the FC domain because we can use Fab. And I'm running out of time, but people have looked for small molecules that bind to DC sign, and they've published this. So we got a hold of 3 compounds that are known to bind to DC sign. We asked, could these compounds inhibit fibrocyte differentiation? So this is effectively a drug screen from a 3-element library, and all 3 worked. And the best one here is compound 1, which has this structure here. It's got an IC50 of 3 picomolar. So for a 3-compound drug screen, this was quite a lot of fun. And then we tried this out in this mouse bleomycin model. And what we can see is that if we do saline or saline plus compound 1, you get this about the same amount of collagen. If you shoot bleomycin into the lungs, you get more collagen. And then 0.01, 0.1 mg per kg of that compound 1, we can basically shut down the formation of new collagen, shut down the fibrosis. So this represents a potential orally available antifibrotic that uses the same receptor, activates the same receptor as that SAP does. Any questions so far? Yeah.
Going back to my original question, have you done that without normal saline, which is can be 10 millimoles higher on sodium chloride than plasma saline?
Oh, shooting that into the lung?
Yeah, so if you're using NS, it's 155 millimoles of sodium chloride.
Mm-hmm.
And from your first earlier slide, you said just elevating sodium chloride by 25 millimoles.
This is,
fibrosis.
yeah, so, oh, I see. So instead of shooting saline into the lung, so there's just 50 microliters of saline into the mouse lung. It'll clear very quickly. And if you just do it with water, or you don't do it at all, you see this is 21 days later. The lung looks completely normal. So now what saline will do, shooting anything down into the lungs, you get some neutrophil influx. And as long as it's not too much of an insult, within about 4 or 5 days, the neutrophils will leave the lungs or die or kill themselves, and the lung will look normal. So the only thing that saline will trigger is a little bit of an initial neutrophil
No one's ever
influx.
tried with 0.45 saline and when the hyponatremia saline.
Oh, I do not think so.
It actually reduces the
No, we've,
response.
no. It's just, but it's with just saline into the lungs, it's just very transient. We've put mice on high and low salt diet and then shot bleo into their lungs, and that's all been published, and I can talk with you about that afterwards. Other questions, comments, insights? So I'm going to try and finish up really quickly. So we showed you that decylating sap seems to affect its ability to calm the innate immune system down. So it turns out we have sialidases, and sialidase inhibitors are potential therapeutics for fibrosis. So salic acid as a background is the distal terminal sugar in lots of glycoconjugates. This sugar is removed by sialidases. These are called neuraminases. So you probably heard of influenza neuraminases like H1N1. N1 is neuraminidase type 1. Influenza and other pathogens have neuraminases. Us and mice, we have 4 sialidases. And I've shown you that SAP inhibits fibrosis, and decylation of SAP blocks its activity. So we just started looking, well, maybe sialidases have something to do with fibrosis. So we can do lectin staining for glycoconjugates that are either sialidated or decylated. And if we look at human patients, patients with lungs that are somewhat damaged, but it's not fibrosis. This is something called COPD. You don't see much decylated glycoconjugates, lots of sialated stuff. But if there's actual fibrosis in the lungs, not much sialated glycoconjugates, lots of decylated glycoconjugates. And if we stain either human or mouse lungs for the 4 different sialidases, so in humans, you see abnormal upregulation of 2 of these sialidase enzymes. And in mice treated with bleomycin, you see upregulation, or 2, or maybe it's 3 of the sialidases. In mice with bleomycin, you see 3 upregulated. These 2 are the same between humans and mice. This and this, number 1 and number 4, seem to show a difference. And if we stain for that one of those sialidases that's common between human and mice, that's upregulated in fibrosis, we're looking at mouse lung fluid. You see elevated levels of this sialidase in the bleomycin-treated mouse lung aspirate, but much lower levels in saline treated mice. And there's a cytokine called TGF-beta, and it seems to be a primary driver of fibrosis. And if we take human white blood cells and we add either, we do control antibodies or stain for TGF-beta, and we treat these with either Nu1 or Nu2 or Nu3 or Nu4, what we see is that, I've mixed this up, I beg your pardon. These are human white blood cells treated with either control or TGF-beta. I'm tired, I'm very sorry, I beg your pardon. And we stained for Nu1, Nu2, Nu3, Nu4. We see that TGF-beta is causing upregulation of these 2 sialidases, Nu1 and Nu2. And TGF-beta also causes human lung epithelial cells and lung fibroblasts to upregulate these 2 sialidase enzymes. Any questions so far? Sorry I'm getting things mixed up, I'm tired. Okay, are we good? Okay. Now, conversely, if we take human white blood cells and we either add nothing or Nu1, Nu2, 3, Nu4, and look at the production of TGF-beta by these cells, we see upregulation of TGF-beta by these 2 sialidases, Nu2 and Nu3. And there's a couple of sialidase inhibitors. One's called DANA, and the other is called Tamiflu. And these can very nicely reduce this sialidase-induced upregulation of TGF-beta. With me so far? So there seems to be this loop. Sialidases are potentiating TGF-beta, and I previously showed you that TGF-beta potentiates sialidase. That was this previous slide. With me? So there's a positive feedback loop between TGF-beta and sialidase. So if there's this positive feedback loop, then maybe if we can shut down sialidases with these small molecule inhibitors, we might be able to shut down fibrosis. So here's lungs at day 21 after treating with bleo or saline. And starting at day 10, when there's already fibrosis, we do daily IP injections of either of these sialidase inhibitors. Here's the lungs treated with saline. Here's the fibrosis in the bleo lung. And bleo plus dana or bleo plus Tamiflu with this injection schedule, the lungs look normal. If we look at quantify the collagen, it looks normal. And so the punchline here is that there seems to be a little bit of a positive feedback loop between sialidases and TGF-beta to sialidases. This seems to be driving fibrosis. And I showed you that sialidases inhibit SAP, and I showed you SAP inhibits fibrocytes. I haven't taken you through the data on the effect of SAP on macrophages, but together sialidases seem to be a nexus for fibrosis, and we can use inhibitors of these enzymes. The sialidase 3 is an extracellular enzyme as a third possible treatment for fibrosis. So there's a bunch of people that did the work. How am I doing on time? I'm 3 minutes over. And this was supported by NIH. The initial SAP stuff was supported by HHMI. And I'm happy to take any questions, and I apologize. I normally don't screw talks up as badly as I did this one for mixing some of the stuff up. But hopefully you got the gist of it. Any questions? Yeah, thank you. Thank you.
Questions?
You good? Yeah.
Yeah. So, if I remember correctly, according to my notes here, so when you were treating with SAP, right, or I don't think you showed data for this, but you alluded to the fact that SAP can skew inflammatory macrophages more towards regulatory macrophages, and I assume that has something to do also with the blocking of the fibrosis pathway. I imagine that would be more associated with inflammatory macrophages.
Yeah, so one of the new concepts in fibrosis is that you can have sort of an M1 driven fibrosis, and you can have an M2 driven fibrosis. You go too far in either extreme. And if you can just bring everything back down to calmness and happiness, that will shut down. And so SAP will skew both of them.
Okay, so mechanistically then, how does SAP influence the skewing towards the regulatory macrophage rather than the inflammatory macrophage? Is that known?
There's some amount is known about the Fc gamma receptor signal transduction pathway, and some is known about the DC sign signal transduction pathway. I'm not terribly interested in agonizing through too much of the signal. We do signal transduction pathway stuff in DICTI, but here I think what's basically happening is that the SAP is a signal. You know how you're a macrophage person, right? Innate.
Not at all, actually, but I'm somewhat familiar with them. Yeah.
Okay, so there's just all kinds of different macrophages, and I think to anthropomorphize, SAP is mostly a blood protein. There's a little bit gets into the tissue, but it's mostly in the blood, and it says, look, you're in the blood. Don't be pulling any weird shit in the blood. I want you to just calm down. You get out into the tissue where you don't see much of me, have at it, go ape. But in the blood, I want you on your best behavior. And that's as good as an explanation as I can think of. Does that make some sense?
It does, and then if I can just have one more quick follow-up then. So do these mice that see that you're treating with extra SAP, or even patients, are they more prone to infection then because they're kind of shutting down that inflammatory response, or is that?
The only side effect that went up with SAP was the patients on SAP. There was a little bit more of them reported coughing, but in terms of adverse events, in the control group there's about 10% adverse events, and the SAP group was about 7%. So it was less. And there's a bunch of things. If you go to the JAMA paper, there's a whole, they looked at a lot of different stuff. And there's a lot of things that were are actually much better in the SAP treatment. The one thing where I would expect the SAP to not be so good is, you're right, if a patient had some really hideous infection and you really wanted to push everything. But remember, out in the tissues, SAP is sort of calming things down a little bit, but it's not completely shutting down the M1-ness, if that makes sense. It also slightly will inhibit wound healing. So the basic idea is if you had a patient on SAP therapy and they got in a car accident and need to heal wounds as fast as possible, you just take them off the treatment for a while. And if they got something awful like tuberculosis, take them off the treatment for a while.
Okay. Okay, thank you.
Other questions, comments, insights? Yeah.
Just a quick question. I think at the beginning you showed the SAP knockout mice.
Yes.
I was wondering, in those mice, do you have circulating fibrocytes or they're still monocytes?
They're still monocytes. So it looks like you need some positive trigger. So one of the things that's been known for in the fibrosis field is that if a hard surface will trigger a lot of the profibrotic fibroblast differentiation, and we think a hard surface trigger, such as plastic, will trigger fibrocyte differentiation. So as long as you don't have some very specific trigger, everything is fine. So you need an additional thing to potentiate. But just for instance, in the bleomycin, we just use a tiny little bit of bleomycin, and that you get this rampant fibrosis. So the answer is no. It needed another signal to drive the fibrocyte differentiation, thankfully. Other questions, comments, insights?
Thank you very much.
Thank you. Thank you very much.